So hello everybody. So I will talk about hepatic in vitro 3D cell models as a novel approach in genetic toxicology. Actually, according to the European legislation, REACH legislation, all the chemicals that are on the market have to be tested for the safety in order to protect human health and environment. This is usually done by a tiered approach, meaning that in the first step, we test the chemicals using in vitro approach in bacteria. When we say safety testing, we are talking here about genotoxic activity of chemicals. So genotoxicity is one of the endpoints that has to be tested for the chemicals. So in the first step, we are using bacterial models. It is called AIMS assay and detects mutations. And then in the next step, we go to cellular models. And we use here the micronucleoside or the chromosomal aberrations. So we test the changes on the chromosome, structural or numerical. If both assays are negative, then the chemical is considered as a negative. And if both assays give a positive result, then the compound is considered as a positive. If we have different results in both tests, then we have to proceed with in vivo, so animals. Usually when we test the safety, we also, let's say, use liver cells, also lymphocytes can be used. And many times the results we get are misleading because they are positive results. Positive results are misleading. 80% of positive results are actually misleading because the cells do not adequately reflect what is happening in our body. And due to this, many compounds that are developed are banned from development, or the in vivo follow-up is done. But because these results are, as I said, misleading, many animals are killed unnecessarily, which is not in the compliance with the 3R. So what is actually 3R? I assume you know that, but I will just summarize it. So the first one is the replacement of animals. Then we have a reduction of animals, and we have a refinement of animals. So 3Rs. On one hand, we can have full replacement of animals. So it means that we completely avoid the animals in research. And we have new technologies that we use, like 3D cell models, organs on chip. Then we have some computer models, so in silico modeling, and some cell tissue engineering that in all these support full replacement of the animals. So like you can see it here. Then we can have also partial replacement, which means that we use organisms that do not experience pain. This is based on the scientific knowledge. And here, for example, we include like a zebrafish model, Daphnia model, and similar. Then we can talk about reduction. This means that we use smaller number of animals for the experiments, and all the experiments are really designed in advance that we use as much as possible out of the every animal. And we also use the correct statistical analysis evaluation, and of course we share the results from each animal. And then the third R is the refinement. It means that housing and experimental conditions are being constantly optimized, that the animals don't feel or minimize the pain, discomfort, and distress. So this means that we always choose the most animal-friendly experimental method, and we promote non-invasive procedures like taking blood and don't killing the animal. So just to mention, that's you. that humans are mostly exposed to 3 different routes. So it's an oral route of exposure. This is by ingesting, so eating food, drinking, pharmaceuticals, you can see it here. And the main target organ is the liver. Yeah? We can have the inhalation route, so by breathing air. This is polluted air, for example, smoking, some cleaning products from the exhaust fumes from the cars. And here the target organ is the lung. And then the third main route of exposure is the dermal, where we can be exposed due to clothes we are wearing, some cleaning products, pharmaceuticals, and similar cosmetic products. And here the skin is the main target organ. So I will be today talking about this one. So the models that can replace not replace, but mimic the liver. Yeah. But before I do this, I will just touch a little bit also the other 2 models. For example, and I'm talking about in vitro systems. So it's not animals, it's in vitro. Yeah. 3D skin models are actually the most developed in vitro 3D models. Why? Because in 2004 and 2009, the EU legislation prohibited to test cosmetic products in animals. Yeah. So first it was the products and also the cosmetic ingredients. And therefore, the cosmetic industry started to develop these models that could better predict and mimic what is happening in our skin. And as an example, here we can see these are small pieces of skin that are actually developed from human primary cells. Yeah. Usually these are fibroblasts and keratinocytes that are grown in the layers. And these these cells are obtained actually at, let's say, plastic surgeries, at some, mostly in the, when you have a beauty, procedures, abdomen, for example, is the best because we have a large surface there. And of course, the companies that get the material and the cells, they have all the ethical approvals, and then they isolate these cells and culture them. And this is called ALI, air liquid interface model, where we have part of the model is in the wet and part is in the air. So it is the combination. And here is just to show you that in these models, we can actually have all the layers from epidermis, dermis, and subcutaneous tissues. Mostly these 2 are developed. So in the skin biopsy, we separate the keratinocyte culture and fibroblast culture. We grow the cells separately. And then when we construct the 3D models, we first take the fibroblast, culture them, mature them for 14 days, and then we add keratinocytes, and you get this model as shown in the previous picture. And this model is actually not only this one, but similar models are used for testing all the cosmetic ingredients. Then in lung model, this one is more simple model. You can also see here that we have co-culture of different types of cells. They are grown on the inside. This is the plastic surface. And on one side you have endothelial cells, then you have epithelial cells. Macrophages are added to stimulate the immune system. And also this system is also called ALI, so air liquid interface, because here we have mostly air. Sometimes we can also have a really tiny liquid surface on the upper part of the model. Okay, but as I said, no, let's go back to the safety assessment of chemicals. Where we mostly use, as I said before, in vitro test system, bacterial, and then also mammalian cells. And I will focus on mammalian cells. Usually, for example, in our laboratory, we are using hepatic cells. Yeah, liver is the main metabolic, the organ that is metabolizing the xenobiotic substances. And also in these cell lines, we want to have metabolism. But the truth is that these cells that are available in research, they inadequately express the metabolic enzymes that are activating and detoxificating the xenobiotic substances. And therefore, we have these misleading results. So what would be the solution? We have to use better metabolically competent in vitro models. That would, of course, resemble in vivo situation, what we actually get in the real life. So 3D models could be this possibility because they exhibit higher level of liver-specific functions, including metabolic enzymes. So here we see the 2D models that are traditionally used in genetic toxicology and cell biology as well. And here are these new advanced 3D models. Okay, so one possibility is human primary hepatocytes are actually still considered as the gold standard in many research areas. Why? Because they are of human origin and they express metabolic enzymes that are very relevant for human exposure and metabolism of xenobiotic substances. But they have also many disadvantages. For example, they have limited availability. We can get primary hepatocytes only at, for example, surgeries when some cancerous tissue is replaced, and they always remove also some healthy tissues. This is the possibility. Or we get material liver from when there is an accident or shooting or something, then the donors that cannot be donors for transplant we can use this material for such purpose, for research purpose. But there is huge genetic and metabolic differences between donors, so due to polymorphism. Also, if we want to isolate and culture the cells, it is a very demanding task, complex task, and the expansion of culture is not possible because the primary human hepatocytes do not divide in cell cultures. And this is also connected to the short lifespan. The cells in vitro situations, so on the plastic surface, they very rapidly differentiate and lose the many hepatic functions and hepatic phenotypes. So eventually, this model is not good for studying the effects of xenobiotics. And everything what I mentioned here is, of course, connected to the high cost of the performance. So what we can say is that human primary hepatocytes already they are considered as golden standard. They are not suitable for routine use for genotoxicity. Yeah, please.
Yeah, I was just wondering, the genetic and metabolic differences, it's stated like a disadvantage, but is it also not an advantage because we have the differences in the population? So
yeah, it depends on what you want to study. Yeah. So I'm talking about genetic toxicology. So for the safety assessment of chemicals. So we want to have reproducible results. So we want to check whether the result we have is really the result reflecting the activity of the chemical. Yeah. And okay, you're true. You're right. It can be advantage or disadvantage. Usually, when we use primary hepatocytes in research, we get the pool of many different donors for 10 donors that are mixed together. And then we have like an average. But still, you can have one donor can, for example, react to some compound or not. Yeah? And if you accidentally get that one that is not reacting, yeah. So what you have to do if you use primary hepatocytes, you have to get the pool, or you have to test the chemical in many different donors. So it was meant in this sense, that because we have like variety of like a polymorphism, it can be a disadvantage here. And usually we want to have repeatable results that also that each laboratory can reproduce. Yeah. Okay, so in history, in past, it was thought that the Earth is flat, but we know that it's spherical. So maybe the model that we are actually traditionally using is not the optimal model, and we should just change the model, and maybe we get more predictable results for human exposure. I'm not sure how much you know about the cell biology, and so I will just a little bit explain. When we have a hepatic or any other type of cells in two-dimensional, so they are actually grown in the surface of the plastic. They grow, we say it, in monolar culture. So the cells have the contact with each other cell only at the limited area. So the communication is very poor between the cells. They have flattened, stretched morphology. And due to this unnatural morphology, actually many cellular processes are affected and they do not reflect the natural end result. And because of this, they also not adequately mimic in vivo microenvironment. On the opposite to this, in 3D models, as you can see, the cells grow in 3 dimensions, and they are virtually 100% surrounded with other cells. When they grow like this, they start to produce their own extracellular matrix. So we don't need to add some matrix. And maybe I just forgot to mention here that the cells here are actually, when we expose them, they are uniformly, equally exposed to molecules or the nutrients, and they all at the same extent can also remove the CO2 or the products at the same extent. But here, on the opposite, we have different zones, or we have this gradient. On the surface, the cells are exposed, they have more access to nutrients and oxygen. And we go toward the inner part, we have less nutrients, less oxygen, but we have here the highest concentration of CO2 and waste products that are reduced toward the outer part of the spheroid. Also, in such a spheroid, we have different zones. For example, here we have proliferating zones where the cells do proliferate. Then we have a question zone where the cells stop to proliferate. They are just there and waiting for the activity. And then in the middle, we have this hypoxic core because of the conditions I mentioned before, high gradient of CO2, waste products. And in genetic toxicology, we actually don't want to have this hypoxic core when we use the model for testing the chemicals, because hypoxic core means necrotic cells, so bad cells. Yeah. And in such a model, we can also co-culture different types of cells to mimic more properly the organs and the in vivo microenvironment. Maybe one advantage of such model is that we can use the model for prolonged exposure. As you will see later on, the cells can stay viable in such conformation for several weeks, even several months. Okay, just to summarize, what are actually the advantages of in vitro 3D hepatic? I'm talking about hepatic models, but actually other cell types can be grown in this conformation as well. So the cells have improved cell and cell matrix interaction. The cells are highly differentiated. The cell morphology and cytoskeleton architecture is more closer to liver cells. They are also express the markers of differentiation and polarized hepatocytes. And very importantly, they have increased level of liver-specific function, which is important in our research studies because we want to have metabolically competent cells that we activate and detoxify the xenobiotic substances. So they express liver-specific genes and proteins, of course, that are involved in xenobiotic metabolism. As I mentioned, they allow prolonged exposure, and they, very importantly, also reduce the experiments on animals. So this is in compliance with the 3R strategy. Probably such models will not replace the animals, but they will reduce the number of animals because we can do a lot of experiments, preliminary experiments in such better models that are than the 2D models traditionally used. So what we can say that these 3D cultures represent the bridge between 2D cultures and the animals. There are different techniques for culturing the cells. They can be varied from a very, very simple spontaneous cell aggregation. Then we have liquid overlay cultures, hanging drop method. This one is very important. It means that the spheroids are formed in drops. They are hanged on the plastic on the opposite side, and then the cells grow in the drops of the medium. Then we can use the force floating method. We can have some microcarrier beads so that the cells are stuck to the different beads. And then these are more or less static conditions. I will explain more later on. But then we can have also some dynamic conditions of culturing, like spinner flask cultures or rotary cell culture systems. They are even more advanced. Or we can have also some matrigels, hydrogels, scaffolds, where the cells stick to the surface and they produce three-dimensional cultures. But they're grown on the scaffolds, like you can see here. So some material that the cells stick to the material. So this is where we have a monolith culture, and then we go to the 3D cultures. So like a scaffold, spheroids, organoids. Spheroids are usually made from the permanent cell lines. Organoids, they can be made from the stem cells and tissues, primary tissues. Then we have scaffolds, as mentioned, and some hybrids like air-liquid interface cultures and organs on chip. So there are many techniques, as I mentioned, but what is the challenge in our research is to have some technique or method that should be simple, reproducible, and should be able to produce many spheroids that we can use them for the high throughput method. For example, in our laboratory, we are using two different techniques. One is the force floating method, where we use centrifugation force to form the spheroids, the aggregates, and then we culture them in the 96 well plates. So I don't know if you see here, but in each well we have a small spot, and this is one spheroid. So as I said, and then the other method is when we grow the spheroids in bioreactors. This is a dynamic system. You will see it later on. So with the force floating method, as I mentioned, the spheroids are formed in the 96 well U-bottom well plates, where the cells do not stick to the plastic. Then how we do this, we count the specific number of cells, and then we put them in the methyl cellulose. This is some matrix we put that the cells stick together. And then we put in the centrifuge that all the cells gather in the middle. after that, we can culture them in the incubator. So counting the cells, the methyl cellulose for the matrix, 96 well plates, each one is one spheroid. So we mix the methyl cellulose and the media with the cells. So this was just for you to get an idea how we do it. At the end, you saw grow them for 3 days. Actually, we can grow them for up to 2 weeks. What we did, we counted the cells and we seeded or we put in this initial culture a different amount of cells. Oh, we did it from 500 to 15,000, yeah? But I will show here only the cells with initial density of 3,000 cells per spheroid and then 6,000. And these I will call small spheroids and these I will call larger spheroids. Yeah? So we left the spheroids. So these are actually the aggregates. We call them aggregates because it's just aggregation of cells. So they are together, but the connections are not formed, yeah? And then we left them in the, wrong button. We can leave them for several days. Here we see up to 3 days, and we can measure the surface area, like here. And by this, we follow monitor the growth of the spheroids. And you can see that here in small spheroids, in small spheroids, we have a nice growth up to 7 days. In larger, there is a growth, but it's not so steep. Now you see that the spheroids grow, but they are denser. And already in the middle, you can see that really there is something happening. Okay. So the other possibility. Okay, so here's something mixed, but okay, it doesn't matter. So this is a dynamic conditions we grow in the agri-well plates where we get these aggregates, yeah, and then we put them in these bioreactors. And here in this image, you can see these small dots. Every dot is one spheroid. Yeah, we can have in such bioreactor we can have up to 500 spheroids. So these spheroids even have better conditions because they grow in dynamic conditions. You will see it later on. I think that I have some slides here mixed, but it doesn't matter. We can grow them for several months. At the end, these spheroids are of the same shape and the same size. And they have, because of this rotation, they have all the time the supply of nutrients, and because faster moving, these nutrients can reach the inner part of the spheroid, and at the same time waste material can go out. So they have better conditioned cells for growth, and they are much more viable. And particularly what we don't need is to add extracellular matrix. I mentioned in the static conditions we have to add methyl cellulose for sticking the cells together, but here this is not needed. Okay, so at the beginning I mentioned that we have here some necrotic core usually formed, yeah, and we have to see, okay, what is happening in our system? Do we have this necrotic core because we don't want it? Yeah? And now I'm back to these smaller and larger spheroids. So this is for the 3,000 initial density, and here is the 6,000 initial density of cells. And you can see, I'm not sure if you see it, but here after 12 days, there was not much red color. Red color is for the propidiomiodide. It entered the dead cells, those that have a permeable membrane. So we assume these are dead cells. Yeah.
Sorry, if I may add, can you look at the TV? It's better seen there.
Yeah. Yeah. Okay, so but I will show it here. Okay. Maybe on both sides. So here, no propidiomiodide, and this FDA stains the viable lifestyles. Yeah, so you can see that maybe we can have some imagination, but not much of dead cells. While in 6,000 cells, so this is these larger spirits, we can, after 6 days, observe some maybe red necrotic core. And when we measure the fluorescence, we see that in these small, up to 12 days, there were less than 20% of dead cells. While in 6,000 initial density, already after 6 days, we have like 20% of dead cells, but this is still not much. So every model has to be optimized before it is used. Okay. So I know that you have, I think, Friday or something, you have some lecture about the flow cytometry, so I will not go into details in the methodological part. We use the flow cytometry for measuring the cell cycle. You know that there are some phases, for example, this is G0, G1, which is the growth preparation for DNA synthesis. Then we have a S phase. This is the G0, G1. S phase, this is for the data application. And then G2 is a preparation for the mitosis, and this is the growth. So these phases can be actually distinguished with flow cytometry. And what we did, we measured each day how many cells we have in the G0, G1, S, and GM phase. And you can see that in smaller spheroids, there was not much of a difference between the days, up to 18 days of culturing. But in a larger spheroid, you can see that there is an increase in the G0, G1 phase. So we have more cells on the account of reduction of cells in the S and GM, G2 phase. Sorry. So why is this happening? What is this? What effect is this reflecting? Okay. Then in the next step, we again use the flow cytometry and we measure the Ki67 marker. This is a marker that is expressed only in proliferating cells. Yeah? And we can see that with time of culturing, there is a reduction of these Ki67 positive cells. So it means that the cells stop to proliferate. This is in smaller as well as in the larger spheroids. Here, these are all the cells that we counted. In this graph, in this one, you see only the cells that we gated that were in the G0, G1 phase. And you can see that actually in both, also in this, this is, if you remember, only this phase, we have a reduction of Ki67 positive cells. I forgot to mention that this marker is expressed in all stages of the cell cycle except in the G0 phase. The G0 phase is the resting phase in the cell cycle. Okay, so again, these images. When we gated only these cells, you see the blue are Ki67 positive cells. And you see that with time of culturing, this blue is small, each day it's smaller. Yeah? So it means that this increase goes to the account of cells accumulating in the G0 phase, and which is the confirmation that the cells are actually resting, not dividing. And why is this so important? Because these 3D models better reflect in vivo liver conditions where the cells do not proliferate all the time, while in 2D cultures, the cells are proliferating all the time. When they stop to proliferate, we have to reculture them. So this reflects better this in vivo situation. Okay, then very important is to see what is happening on the molecular level. So with the qPCR, we measured the expression of genes. And here you see the 3 different genes. This is topoisomerase 2, PCNA, and Ki67. All these 3 are involved in the cell proliferation. And what you can see that they were all were downregulated. 1 means this is the comparison with the monolayer culture. So monolayer culture after 2 days of culturing, this is usually the time when we use the culture for experiments in 2D. So this lower line, it's the, let's say, the biologically important difference with what we measure the control. So the control here was the 2D monolayer culture, and we can see nicely that all these genes were downregulated with time of exposure. Then we measure also some cyclins that are also involved in the cell proliferation. Again, you can see the huge downregulation, 0.3-fold induction of these genes, and also some. So HIF1A is hypoxia factor induced, and also Puma BBC3. This is the apoptotic, the gene that is encoding the apoptotic protein. So what you can see here, maybe this is not seen nicely, but still you see that also these 2 genes were downregulated. So no hypoxia and no apoptosis is in such models. Very important is the metabolic activity and the hepatic markers in the models. So we measured what is happening with the alpha fetoprotein and albumin. These are 2 very important markers, hepatic markers, and you can see that already after 3 days of exposure, they were upregulated. And maybe at 7-10 days, we see that there is some reaching the plateau. Then here it's a lot of data, but it's not meant that you will see all the numbers. Just to get the idea, again, this dotted line is the comparison with the 2D monolayer culture. And here you see the phase 1, those genes that are encoding phase 1 proteins. These are mostly cytochromes. We know that this is a huge family of different CYPs. And as you can see, all were upregulated in 3D model compared to 2D model, particularly increasing with time. Here I want to pay your attention in the CYP3A4. These are HepG2 cells. I forgot to mention this, but this is the specific cell line that is used in genetic toxicology. And here, this gene, here you see it's very upregulated. In 2D models, we can hardly detect the expression of this gene. And this one is a liver specific. 3A4 is a specific for livers. Then in the second phase enzymes, these are detoxifying enzymes. Important are the UGT, sulfotransferases, and acetyltransferases. These are the most important enzymes. And here you can see that the genes encoding them were again upregulated, or maybe some were downregulated, because there are again several different isoforms, and they can be up or downregulated. But we see that they were expressed in particularly important sulfotransferases that are again hardly expressed in 2D models, but here we can see a really nice expression of these genes. So what we can say is that we have expression of liver-specific markers from the phase 1 and phase 2 metabolic enzymes, and to compare to 2D, 3D models are more metabolically competent. What we then from this data, we started we designed an approach that can be used for genotoxicity assessment or on the other side, safety assessment, which is actually the same methods are used, just it's called differently. So we have cells, we see them, we make the spheroids like this, and then we can expose them to different compounds and treat them for specific time. Here we took like a 24, which is considered as a short time exposure, or several days, which can be like a prolonged exposure, even longer than 72 hours. Then we can measure different endpoints ranging from cytotoxicity. Then we can measure the surface area, and by this we can see whether there is influence of the division and growth of spheroids. And then we can stain with different fluorescent markers. Also, we can apply the Comet assay, the expression of genes, and we conform this with the proteins. And of course, for several applications, we need single cell suspension to obtain from such spheroids, which can be quite a demanding process, and we have to optimize the conditions that we get a viable cells. We don't want to have dead cells, yeah. And then we can use them for a different application in the flow cytometry, where we can actually measure simultaneously several endpoints, because we label the cells with different chromochromes or dyes, and then on different wavelengths we can monitor the markers. And we can go up to 8 fluorochromes different, detecting in the same cell. In particular, if we add also the cell cycling, we can check in which phase of the cell cycle the cells with specific marker are okay. Yeah, so I mentioned that the preparation of single cell suspension is a critical step. So how we do this? Again, we grow the cells, the spheroids, or in bioreactors, depending on the method we have. And then we take the spheroids. We use the cut tip. We don't want to damage the spheroids. And we use the enzymes. Usually we make a cocktail of trypsin and the collagenase, so different enzymes, and then incubate for a couple of minutes. We add the FBS, because by this we stop the enzymes, and then we have a single cell suspension, and we check the viability. I mentioned the Comet assay. Probably there was no lecture on this, but it will be in the next days. So the Comet assay is, it detects double, but mostly single strand breaks, alkali label sites like apurinic, aprimidinic sites, also DNA or DNA protein crosslinks, and single strand breaks that are associated with the incomplete excision repair. And here, maybe on both monitors, you can see it better. We have DNA, so nucleus, with almost not damaged DNA, but here you can see the tail. So in the Comet assay, we say that we have a head and the tail of a Comet assay. So more the DNA is in the tail, more damaged the DNA is. As I mentioned, we have digestion with some enzymes, and then at the end we get these cells, and we can use them for different application, like a Comet assay or flow cytometric analysis. So just briefly explain the Comet assay. We have a single cell suspension. Like here, we edit enzymes. This is the spheroids are treated with some chemicals, and then we proceed the Comet assay. So here, these spheroids are, let's say, already treated with our compounds that we are investigating. Yeah. So we want from these spheroids get single cells. As I mentioned before, we use the enzymes, and then we put this on the agarose gels. We do the lysis at a high pH with detergents, 16 minutes, and then we do the unwinding and the electrophoresis. And after that, we have a neutralization at the pH 7.5, and then we stain them with different stains, to get all the stain only the DNA. So in these phases, we get rid of all the proteins, histones, only the naked DNA stays. Yeah. And then at the end, we can analyze these are the comets. You see better, but I'm sure that there will be lecture specific for this, so I will not go into details. I just wanted to show you that you better understand what we did. So we compared the effect in 2D culture, in the monolar, and in these spheroid cultures. We exposed to different compounds for 24 hours, for example, and of course we checked first the viability. Cytotoxicity has to be checked before we check the genotoxicity, because we don't want to test genotoxic effects on cytotoxic concentrations where the cells are dead. So then we performed the comet assay, and the model genotoxic compounds we selected were benzoiparin. This is the compound that is formed in the incomplete combustion. Aflatoxin is a mycotoxin, so the fungi form it. It's a natural toxin. And then we have two FIPIN-IQ are two heterocyclic amines that are formed at high temperatures in grilled meat and fish as well. And etoposide is a cytostatic drugs, which is a direct acting compound. Okay, so if we compare in 2D models, you have first is always the 2D model and then a 3D model. You can see that we detected lesions, DNA damage at the lower concentrations in the 3D model. So the low concentration was detected. Then in aflatoxin, actually there was not such difference between 2D and 3D model. In both systems, we got a similar result. And then these heterocyclic amines I mentioned. In 2D models, as you can see, in both cases, we did not detect DNA damage, but in 3D models, we did. So where could be the reason? In these heterocyclic amines, very important are sulfotransferases. So it means that these enzymes are not expressed. And we saw before, if you remember, that in 3D models, these enzymes, at least at the mRNA level, they are expressed. Yeah, so this is then the result that the 3D model was more sensitive. And then in the etoposide, which is a direct acting genotoxic compound, both systems detected the lesions that was expected, because we don't need a metabolic activity. Okay, so in this system, after the treatment of spheroids, we also tested some genes that are involved in metabolic metabolism. As you can see, again, we will not go into details, but just to show you that cytochromes and also sulfotransferases and acetyltransferases, they were upregulated in the systems, particularly in FIP and IQ, you can see this. And here this line is the control. So it's not here the 2D, but it's a spheroid control. And this dashed line, this is what is considered as a positive, biologically positive result. So what is above this, it's a positive. It's considered 1.5-fold increase compared to control. Then we also measured some genes that react like as a response to DNA damage. For example, very important are p53. These cells express the p53, which is very important. And then also some downstream regulatory genes, as you can see here in these pathways. These are cyclin, CDKN1A, that is encoding the P21. It's important in the repair and arresting the cells due to the repair. And then GD45 alpha, this is also important one in the DNA repair, and ERCC4, again, important in repair. And actually what we can see is that all except the FIP. Some of the genes were upregulated. So it means that there was some damage, and due to this damage, the repair was activated. And this was again then confirmed at the end on the protein level, because on the protein level it's very important that we confirm the results of what we have at the end. Okay, so I mentioned before that this can be also used for the flow cytometry. We have seen the cells, we label them with different antibodies, different dyes, and we then detect at the laser with the lasers specific fluorochromes. And at the same time, for example, we detect the cell cycling, then Ki67 marker, and also the H2AX positive cells. These are specific for the DNA double-strand breaks. So this is the approach we are also using in the genotoxicity assessment. Here we selected only benzoyperine and FIP, like two model genotoxic compounds. And what is happening in the cell cycle, you see that there is a reduction of the G1 phase and increase of the G2 and S phase. Usually G2 phase is activated, or the cells, it's not activated, sorry, but the cells are arrested in the G2 phase. Due to the repair. When there is a repair, there are the checkpoints for the repair. We have arresting in cells in the G2 phase. So it was confirmed that these cells that are exposed to benzoyperine were arrested in the G2 phase. Very similar also, also after prolonged exposure. Not so obviously was this detected for the FIP after 24 and 72 hours, but there were some differences. Okay, what about the Ki67 marker? So the proliferation of cells, we can see that there was some reduction, but not much reduction of the proliferation of cells. And we detected in both cases with benzoyperine and FIP increase of double-strand breaks. So it means that these two model compounds induce double-strand breaks. So we kind of confirmed that the model can detect these lesions. So this was the purpose. That's why we use the model compounds to confirm the suitability of the model for testing genotoxic compounds. Okay, so before I already mentioned dynamic system. So you can see here. These are, for example, the incubators where we have 6 different rotors, and on each rotor there is such a bioreactor with these dots. These are the spheroids that grow in the bioreactor. So we can actually see with the tablet. On this tablet you see one bioreactor rotating in the incubator, and we can monitor everything. We can change the speed of the rotation we actually don't need to open the incubator. This is one bioreactor that is rotating, but we can use 6 at the one incubator, and for each one we can have different rotation that is adjusting according to the size of this spheroid. So I will not repeat, maybe just that these are the aggregates plates that are used for forming the aggregates during the night. Once we have these aggregates, we put them into bioreactors, and in the incubator, we can grow them for up to several months without a drop of viability, but we actually don't use, we use up to 1 month because there is no need to have such a long culturing. And here then this is the collection of the spheroids. So here you see we monitored what is happening day 1, up to day 27. And you really see that these spheroids grow nicely. The spheroids are the same shape, the same size approximately at the end. And after 27 days, really the images tell by themselves that they grow and even more we could leave the spheroids. So again, in this is now a dynamic system, yeah, different than the static condition I showed before. Again, we monitored what is happening with the cells, how do they proliferate. And here you see that after 6 days of culturing, the majority was in the G1 phase. This is normally that the cells are mostly in the G1 phase, also in the 2D cultures. So with time of culturing, we see that this G1 phase, the cells are a kind of arresting in this phase, and after 21 days, we see here something is happening, and this G0 phase started to occur, and after 24 days, mostly like 80% of cells were in the G0 phase. So these were these resting cells, not proliferating cells. We checked also after 27 days, mostly almost 100% were in G0 phase, so the cells were not dividing anymore. This is also when we measured when we counted the cells, you see here nicely that all these three phases are decreasing the number of cells, on the account of increasing the cells in the G0 phase. This is the red one. Then we also, with the pyronine is a stain that binds to the RNA. So we measured what is happening with those cells that are actually viable and are proliferating, whether we, again, get the same amount, the same result. And you see that here, this is what is happening here. These are those cells that are labeled with the pyronine. So we get, again, the effect showing that the cells are not proliferating anymore. The blue are the proliferating cells that are reducing with time, and then the red are non-proliferating cells that are increasing with time. Again, we confirm that in this system, the cells stop to proliferate. And the third marker, this is the Ki67 marker I'd explained before. Again, we see that the cells are actually that are not, these are Ki67 negative cells that are not expressing this marker. And you see a drop, like a 10% maybe at the end are proliferating. Okay, we also used immunocytochemistry where we I don't know if you see it, but here it's supposed to be the spheroid cut, and these are only the parts of the spheroid. But you can see this is like one zone, and then you have another towards the inner part. You can see that this is the different labeling. These are the cells that are Ki67 positive, so it means that these cells do proliferate, but here we don't see this proliferation anymore. So this is, if you remember at the beginning, I explained that we have different zones, so we showed it here nicely. Then we have also some multi-drug resistant associated protein that are responsible for efflux of the compounds from the cells, and also some cytokeratins, again, different zones labeled. We also tried to apply the Comet assay on such a model, but because we have really dense mature spheroids, we checked first what is the basal level of DNA damage in the spheroids, and we can see that actually there were not much of a damage on the vessel. So we can use this approach for studying the effect of compounds on DNA damage induction. And this was done by culturing the cells in bioreactors for 21 days. We see a nice spheroids form, and then we expose them for 24 or 96 hours to benzoyl and FIP. Again, these two model compounds we are using all the time. This was considered as a short-term exposure, and this is as a prolonged chronic exposure, which is very relevant for humans because in everyday life we can be exposed to really low concentrations of the chemicals and through the longer, like a chronic exposure. So benzoyl after 24 hours increased the damage as seen here, as well as after 96 hours. What about the FIP? The FIP, the other compound, actually after 24 hours we did detect some damage, but not after 96 hours. Then we measured also the expression of genes that are involved in the metabolism of symbiotic substances as before, like cytochromes, UGTs, sulfotransferases. This is without treatment, only the spheroids. Compared to the line is the 2D culture after 2 days of culturing. And we can see that several genes were upregulated, particularly after 26 days. Mostly important are the cytochromes, also sulfotransferases and UGTs. After exposing the cells, the spheroids to these two compounds, we can see that these genes, particularly CYPs, were really highly upregulated in 24 and 96 hours. Here also some second phase enzyme, genes encoding enzymes, and as well as in the FIP. Here also UGTs, and here some sulfotransferases. Then we measured also the genes that are differentially expressed due to the response to DNA damage. And we can see that in benzoyl they were upregulated, as well as in FIP after 24 hours, but not after 96 hours. And if you remember, I showed in the COMET assay, we didn't have the effect on the damage. So it means that by that time, probably damage was already repaired, because this is also a possibility to see the COMET assay, the repair of the damage. After, let's say, optimization and to see how the model behaves, we can say that this 3D cell model shows a high metabolic activity and can be used for detecting genotoxic compounds. Maybe also one interesting thing. I showed at the beginning these 3 layers. What we can also do, and of course, in these layers, the cells, they behave differently, yeah? And we can separate, in mature spheroids, we can separate these layers. And here is the outer part, and this is the inner part. So we can, get 2 layers. And of course, we can check what is happening in the cells here and also here. How we do this? We take the spheroid, we put in some enzymatic treatment, but for a shorter time, and then with pipetting up and down, we disintegrate the spheroid, and then we get the cells from the core and the cells from the rime, and we have separated population. So at the end, this is just a summary of the methods of the approach we are using. So from the monolayer culture, we perform in the agribase the aggregates. We put them in the incubator, in the bioreactors, And then with these bioreactors, we can treat the spheroids with different compounds. And then we check the different endpoints from the cytotoxicity, measuring the planimetry of the growth. With the confocal microscopy, we can check different markers. Then with transcriptomic and proteomic analysis, immunocytochemistry as shown. And then, on the other hand, we can disintegrate the spheroids to the single cell suspension by enzymatic treatment, and we can perform the comet assay, the micronucleus assay, as well as flow cytometric analysis of different endpoints. So at the end, just to conclude, that these advanced hepatic in vitro 3D cell models are very metabolically competent if we compare them to traditional 2D models. And they represent a very promising model for the safety assessment of various compounds. We can use them also for mechanistic studies, investigating genotoxic hazard that is posed by the chemicals, food products, environmental pollutants, and also complex mixtures that are important topic to study because humans are not exposed to single compounds, but we are exposed to complex mixtures in the everyday life. Also, these in vitro 3D cell models, they can be used to fulfill the 3R strategy, so reduce, replace, and refine the animals. And at the same time, they also avoid this inaccurate prediction of animal models to human exposure, because animals that are mostly used are rodents, and rodents are very far from the humans, so it's only the prediction. And here we are using the human cell lines. So as I mentioned before, probably these 3D models will not replace the animals, but they for sure bridge some gap between the 2D models and the animals. And of course, it's important to further validate these models. So this is my team, my researchers, students that were involved in this huge amount of experiments. Also, the part of the work was done in the collaboration with the Danish researchers from the Southern Denmark University. So the part of these results is also done within the Horizon Europe Cat Cancer project. Of course, thank you for your attention.